Welcome back to Biology 2225, AMP2 at CNM. We are discussing the urinary system, Chapter 23 in our textbooks. We left off last time at objective number 78. So what we're talking about is the regulation of glomerular filtration rate. And we said that there were going to be three different mechanisms for doing this, what we call renal autoregulation, the sympathetic nervous system control, and hormonal control. So we've just completed talking about the renal autoregulation mechanisms, and there were two. One was called the myogenic mechanism, where the afferent arteriole simply constricts in response to high blood pressure, thereby reducing pressure and flow in response to high blood pressure, so that the filtration rate doesn't go up automatically when blood pressure goes up. The second mechanism was a little bit more complex, involving what's called the juxtaglomerular apparatus, and that was a complicated set of structures known as macula densa. The macula densa secretes ATP, remember, in response to increased sodium and chloride in the tubular fluid. Increased sodium and chloride in the tubular fluid result from high glomerular filtration rates. The reason that is, remember, is because the higher the glomerular filtration rate, the faster the fluid moves through the tubules. And so sodium and chloride don't have as much opportunity to be reabsorbed with that faster flow. And so by the time they reach the beginning of the distal convoluted tubule, where the macula densa cells are, the sodium and chloride is usually in higher concentration. The macula densa cells sense this higher concentration and secrete ATP. Sitting right next to the macula densa cells in the wall of the distal tubule and distal convoluted tubule are mesangial cells, a little patch of cells right next to that tubule. They pick up this ATP, remember, and they convert it to adenosine. Adenosine then travels to a special group of cells called granular cells, which are also right next to these other two types of cells. But these granular cells, recall, are specialized smooth muscle cells wrapped around the outside of the afferent arteriole. The adenosine will stimulate those granular cells to contract, causing afferent arteriole constriction. This reduces blood flow and blood pressure into the glomerulus, and that reduction in pressure is going to reduce the glomerular filtration rate. So we started with a higher than normal glomerular filtration rate, and we see that as a result of this complex mechanism, we bring it back down. And so that's where we left off last time, and you would think that those two mechanisms alone would be enough to regulate glomerular filtration rates, but renal autoregulation, these two mechanisms can only account for minute-to-minute small adjustments. They can't account for chronic issues or very large changes in pressure, so we need a couple other mechanisms to help us out. So the second overall mechanism that helps regulate glomerular filtration rate is nerve-controlled, specifically sympathetic nervous system regulation. Sympathetic nerve fibers, as you can see here, innervate the renal blood vessels. This includes the major vessels, but also the tiny vessels, such as at the arterial level. Remember, the sympathetic nervous system releases norepinephrine as a neurotransmitter to its target. And usually, when we have a sympathetic response, that also involves the adrenal glands releasing the hormone epinephrine as well into the bloodstream. And so we get usually both norepinephrine from the sympathetic nervous system and epinephrine from the adrenal gland arriving and stimulating the afferent arterioles at the same time, or first the nervous system, of course, because it's faster, and then the hormone follows up. Both chemicals affect the afferent arterioles of the kidneys by causing constriction. And of course, once again, constriction of the afferent arterioles is going to reduce blood pressure and blood flow in the glomerulus, and that's going to reduce glomerular filtration rates. And so the sympathetic nervous system, the ultimate end result is that it reduces glomerular filtration rate, which is essentially what we want during a fight or flight response, or even something as basic as exercise, because we need blood in the heart and the skeletal muscles during exercise. And so we want to redirect blood flow away from the kidneys, and of course that reduces urine production, as we've said before, happens during exercise. And this can reduce, as you can see here, this can reduce glomerular filtration rates to as low as a few milliliters per minute. Remember that typical glomerular filtration rates are on the order of 105 to 120 milliliters per minute. And so this mechanism can drastically reduce glomerular filtration rates, at least temporarily. You wouldn't want to do this for a prolonged period of time, but during something like exercise, it does happen. And then finally, remember we have hormonal control as well. Now, technically, we just saw a hormone affect the glomerular filtration rate. That was epinephrine from the adrenal medulla. But there is a whole nother set of hormonal interactions that affect glomerular filtration rates as well. And this is something we've seen before. Again, it's the renin-angiotensin mechanism. We talked about this in the context of regulating blood pressure back in our blood vessel chapter. But now we're talking about it in the context of affecting glomerular filtration rates. And so recall, in response to a drop in blood pressure, we have baroreceptors in the carotid artery and the aorta, of course, that sense that, and that is going to initiate the sympathetic response that we just talked about. But additionally, that sympathetic fibers that are triggered by that drop in blood pressure are going to also stimulate the granular cells. Remember, again, these are the cells that are wrapped around the afferent arteriole. In addition to being able to constrict and reduce flow through the afferent arteriole, recall these are the cells that also release the chemical renin. So, long story short, as blood pressure drops, as we know, we get a release of renin by the kidney, specifically the granular cells. You may remember that renin then converts angiotensinogen into angiotensin 1. Angiotensin 1 then has to be converted into angiotensin 2. Here we see the sequence of events. The precursor angiotensinogen is produced by the liver. Remember, when in doubt, plasma proteins are produced by the liver. Always a good guess. The drop in blood pressure causes the granular cells again to release renin. Renin converts angiotensinogen into angiotensin 1. This is not quite the active form yet. We need another enzyme to convert it into angiotensin 2. And you may remember from our discussion of the respiratory system that this enzyme actually comes from lung tissue. The angiotensin 2 then, once it's produced, you may recall, has a wide variety of effects that are going to affect systemic blood pressure, which of course then is going to affect glomerular filtration rates. It also has a couple of direct effects on the glomerular filtration rate as well. Here we see the wide variety of effects that angiotensin 2 has that affect blood pressure, many of which we've already talked about, but there are a couple of new ones in here that I want to point out as well. So remember, angiotensin 2 is a potent vasoconstrictor. It causes system-wide vasoconstriction, and of course that increases blood pressure. An increase in blood pressure is going to tend to increase glomerular filtration rates if not compensated by some other mechanism. A very specific effect of angiotensin 2 that we had not talked about before is that it constricts the efferent arterioles. It actually constricts both afferent and efferent, but its effect on the efferent is much, much greater than it is on the afferent. And so the end result is that even though we might have low blood pressure in the system, because that's what started all this, remember, we're actually going to get an increase in glomerular filtration rates. Why? Well, by constricting the efferent arteriole, we're allowing less blood to exit the glomerulus. And so as blood trickles in through the afferent arteriole and it can't get out as effectively through the efferent arteriole, that is going to cause the buildup of back pressure inside the glomerulus, and that's going to increase filtration rates. Another interesting thing that constriction of the efferent arteriole does is it lowers pressure in the peritubular capillaries. Remember, the efferent arteriole carries blood from the glomerulus to the second capillary bed in the portal circuit, which is the peritubular capillaries. Well, if I constrict the efferent arteriole, I'm essentially constricting the blood supply to the peritubular capillaries, and that's going to lower the blood pressure. Well, if I lower the blood pressure in the peritubular capillaries, that's going to allow for more reabsorption. Remember, reabsorption is going to be taking place from tubular fluid back into the bloodstream. And once again, movement of fluids between tubules or tissue spaces and a blood vessel is largely influenced by the blood pressure in that blood vessel. And so with lower blood pressure in the peritubular capillaries, I essentially have less force resisting the movement of materials back into the bloodstream. So I'm going to get more reabsorption of salt and water as a result. If I get, again, notice what the effect of that is going to be. If I get more salt and especially water reabsorbing back into the blood, that is going to increase blood pressure. And remember, the initial stimulus for this entire process was a reduction in blood pressure. So again, this is a nice feedback mechanism. Recall that angiotensin II also stimulates the release of aldosterone from the adrenal cortex. As we'll see in some detail later in this chapter, aldosterone promotes sodium and water reabsorption in the distal convoluted tubule and collecting duct. And again, the more sodium and water we reabsorb, that increases blood volume, which increases blood pressure, which is, again, an adjustment to the original problem, loss of blood pressure. Interestingly enough, something we didn't talk about originally is that angiotensin II also itself will stimulate sodium and water reabsorption, but not in the distal convoluted tubule and collecting duct, but in the proximal convoluted tubule. So angiotensin II has a direct effect on sodium reabsorption, increasing it, which causes water to follow by osmosis. Again, increasing blood volume, which increases blood pressure. We also see here that angiotensin II stimulates the release of antidiuretic hormone from the posterior pituitary. And remember that antidiuretic hormone promotes water reabsorption in the collecting duct. So once again, we're going to reabsorb more water out of the tubular fluid back into the blood, increasing blood volume, which compensates for the blood pressure problem, the lower blood pressure that started this whole ball rolling. And then finally, recall angiotensin II stimulates the hypothalamus to give you a sense of thirst, encouraging you to drink. Drinking, of course, introduces water to the body, which will then be absorbed, increasing blood volume, and once again, increasing blood pressure. So most of angiotensin II's activities are essentially a response to a systemic loss in blood pressure by compensating in various ways to increase blood pressure. Of course, indirectly, that's going to influence glomerular filtration rates. Anything that affects systemic blood pressure is going to affect glomerular filtration rate. We also see, though, that it has a very specific effect on glomerular filtration rate by constricting the efferent arteriole and raising glomerular blood pressure specifically. So lots going on with angiotensin II. Okay, so that completes our discussion of glomerular filtration for now. The next unit in this chapter, Unit 4, has to do with the second stage of, or the next two physiological processes involved in forming urine, which are, again, reabsorption and secretion. And so we're going to talk about reabsorption first. Now, if you look at this particular slide, your authors like to break it down into tubular reabsorption, tubular secretion, and water conservation. And they do discuss it in three separate phases. I just want to point out that water conservation is really just a specific example of reabsorption, and that's why I tend to just think about reabsorption and secretion as being the other two processes that occur. Yes, water conservation is important, and yes, it does happen. Generally speaking, we can regulate water conservation tightly using hormones, as we'll see near the end of the process, but we are talking about taking something from the tubular fluid and moving it back into the bloodstream. We're just specifically talking about water, but that is reabsorption. Whenever we take something from the tubular fluid and move it back into the bloodstream, that's called reabsorption. So water conservation, that last process here, is again just a very specific type of tubular reabsorption. All right, so where is all this going to happen? Well, again, if we straighten out the nephron so that the proximal and distal tubules aren't all convoluted, we see, generally speaking, what's going to be happening as a preview is we're going to get tubular reabsorption occurring actually throughout the nephron. It shows it happening here mostly in the proximal convoluted tubule, and it is true that most tubular reabsorption, about 65%, takes place in the proximal convoluted tubule, where things will be moved again from the tubule into the peritubular capillaries. But don't let this diagram confuse you. There is also going to be some tubular reabsorption occurring in the loop of Henle and some in the distal convoluted tubule and collecting ducts as well. Here again we see water moving out of the tubule eventually into the capillaries. So again, this is an example of reabsorption as well. Tubular secretion tends to happen most in the distal convoluted tubule, but it does also happen in the loop of Henle, and those are probably the two main places where secretion takes place, as we'll see. All right, so we're going to talk about reabsorption first. And we'll start, of course, with what happens in the proximal convoluted tubule. As I mentioned, most reabsorption takes place in the proximal convoluted tubule, about 65%. And again, with reabsorption, we're taking substances. From the tubular fluid and moving it back into the blood. Notice, as I mentioned just previously, there is a little bit of secretion that takes place in the proximal convoluted tubule, but most secretion takes place in the distal convoluted tubule. But again, some does occur here in the proximal convoluted tubule as well. Recall that the simple cuboidal epithelium that lines the proximal convoluted tubule exhibits lots of microvilli, again dramatically increasing the surface area, which of course is going to benefit movement of materials across the membrane, which again is reabsorption and secretion. So microvilli, as always, represent a dramatic increase in surface area. Much of this movement of reabsorption especially, but secretion as well, is energy requiring. We're going to have active transport taking place. Not all of it. Some of it is going to be driven by diffusion and osmosis, but much of it, if not most, has to be carried out by pumping materials across the wall of the proximal convoluted tubule. And so, not surprising to find that these cuboidal cells have lots of mitochondria involved in producing the ATP required for active transport. And again, it is a significant energy expenditure in the body. This is an interesting stat right here. The proximal convoluted tubules of your kidneys by themselves account for about 6% of your resting metabolic rate, your resting consumption of ATP. Remember from AMP1 that much of your basic metabolic rate is burning of ATP in muscles to maintain muscle tone. The sodium-potassium pump, which is active transport pump, uses a lot of the body's basic metabolic ATP. But the proximal convoluted tubules also represent a significant portion of what we call your resting metabolic rate. Here we see electron micrographs again of the proximal convoluted tubule, and the most interesting thing to note again is this little brush border here of microvilli. Right, this little fuzzy patch. You may think these are cilia, but these are actually microvilli forming a brush border. And this is very high magnifications with very good resolution, so you can actually see the microvilli in some places. All right, so how does reabsorption take place? Well, as we see, there's two different routes, two different methods, really, mechanisms for reabsorption. One is called the transcellular route. The other is called the paracellular route. The transcellular route is when substances move through the cuboidal cells lining the proximal convoluted tubule. And by through, what I mean is they have to pass through their microvilli, the membrane of the microvilli on the luminal side, pass through the cytoplasm, and then pass out of the epithelial cell on the other side, the basal lamina or basement membrane side. This is the route that is somewhat selective, of course, because anytime you pass through a membrane, you have to deal with the selective permeability of that membrane. And this is the route that would be involved in any kind of active transport, because remember, active transport is carried out by energy-dependent transport molecules that are embedded in cell membranes. On the other hand, the paracellular route occurs when substances pass between the cuboidal cells of the epithelium of the proximal convoluted tubule. And so even though we have tight junctions between the epithelial cells, small materials can still leak and squeeze their way through in between, and that includes water, for instance. So this of course would be a passive process. There's no ability to use ATP to pump or selectively transport materials this way because they're simply leaking between cells. So this tends to be an osmotic or diffusion type of process. And so water moves especially well through this paracellular route. And recall, when water moves, it tends to drag solutes with it. This is referred to, remember, as solvent drag, and we've discussed that before. Regardless of how the substances make it through, either through the transcellular route or the paracellular route, eventually they end up again on the basal side of the epithelium near the basement lamina, and right next to that, of course, would be your peritubular capillaries. And so the materials and the fluids that make it through end up ultimately being picked up by the peritubular capillaries, so they're returned to the bloodstream. And again, this is essentially the point of reabsorption. It's getting materials back into the bloodstream. Now, sodium is a key substance to be transported, because as sodium goes, so do many other things. Why? Well, as sodium is reabsorbed, of course, that creates osmotic imbalances, which then will cause osmosis of water to occur. And then, as we just said, water, as it moves, will tend to drag a lot of other solutes with it. The other thing that transport of sodium does is we can also pump it and create not just osmotic gradients, concentration gradients, but we can also create electrical gradients. And so as sodium is pumped through, we create electrical imbalances between the tubular fluid and the fluid that's on the basal side of the epithelium, and that can attract anions like chloride across the epithelial lining. And so sodium reabsorption really is at the heart of the movement of a lot of other materials. Remember also that sodium is the most abundant ion in the blood, and so not surprising to find that it's the most abundant ion in the filtrate as well. And so as we pump sodium, we really do affect significantly osmotic and electrical gradients. So how do we take up sodium, which then leads to the movement of many other things? Once again, there's two types of transport proteins in the apical surface of the luminal surface, the side that has the microvilli of these cuboidal cells. There's two types of sodium transport proteins. The first one is a symport transporter. And remember, a symport is a molecule, a protein embedded in the membrane that transports two separate kinds of materials at the same time. And specifically, this particular symport is going to be a molecule that binds sodium and some other solute. So there are different types of symports. Some of them bind sodium and glucose. Some of them bind sodium and amino acids. Some of them bind sodium and lactates. And we actually have three different symport molecules, each designed to transport sodium with another type of molecule. A symport always moves the two materials in the same direction. Remember, an antiport transporter transports two molecules in opposite directions. Symports transport them in the same direction. And so these symport transporters are going to pick up a sodium ion and then one of these three other molecules, depending on what kind of symporter it is, and it's going to transport them both into the cuboidal cell. The other kind of sodium transport protein we have in the apical surface of the cuboidal cells is a sodium hydrogen ion antiport. Remember, again, antiport, opposite directions. This transporter is going to pull sodium into the cell, and at the same time, it's going to pump hydrogen ions out into the tubular fluid. So we're reabsorbing sodium essentially, but at the same time, we'll be secreting hydrogen ions. Again, remember, hydrogen ions are the ions involved in determining the pH of a fluid. And so this is an important element in the kidney's job of regulating blood pH. And you see it is tied to sodium transport. Now, once the sodium arrives inside the cuboidal epithelium, it begins to accumulate. Of course, its concentration begins to rise, and of course that would make it difficult for more sodium to continue to diffuse into the cell as the sodium concentration rises inside the cell. It also makes the interior of the cell more and more positive as it accumulates, which will disrupt the membrane potential of the cells. So we can't let the sodium that gets into the cuboidal cell just sit there and accumulate. We have to move it out so we can maintain that positive, that favorable concentration gradient, so sodium keeps moving into the cell. Well, remember, all cells have a sodium-potassium pump. Excuse me. And these cuboidal cells that line the proximal convoluted tubule have their sodium-potassium pumps localized in the basal side of the cuboidal cells. So on the apical side, facing the tubular fluid, they don't have any sodium-potassium pumps, and sodium only moves into the cell. But on the opposite side, the basal side, that's facing, essentially facing the peritubular capillaries, we have lots of sodium-potassium pumps. And remember, the sodium-potassium pump always pumps sodium out of cells. And so on the basal side, the sodium that entered the apical side is going to get pumped out by the sodium-potassium pumps into the extracellular fluid around the basement lamina or basement membrane. And of course, the peritubular capillaries, as we said, are right there. And so they will pick up all this sodium that is now accumulating in the extracellular fluid. They simply pick it up by simple diffusion, and now the sodium has been returned to the blood. So notice the energy for all of this movement of sodium is ultimately being supplied by the sodium-potassium pump. Remember, these are active transporters. These require ATP. And of course, the sodium-potassium pump pumping sodium out into that extracellular space creates the concentration gradient that then drives the sodium into the peritubular capillaries. The sodium-potassium pump also creates the low sodium concentration inside the cuboidal cell that creates the concentration gradient that then helps draw sodium in from the tubular fluid in the proximal convoluted tubule. And so the energy created by the energy used by the sodium-potassium pump to create these sodium ion gradients is what drives the movement of sodium into the cell and the movement of sodium into the capillaries. This, you may recall, is an example of what we call secondary active transport. Secondary active transport is when we can transport materials, sometimes against their own concentration gradient, but the energy used to do that was actually spent somewhere else. In this case, the energy is used by the sodium-potassium pump, and then that drives the movement of sodium elsewhere in the body. In this case, the apical surface of the cell and into the peritubular capillaries. Remember, we said that since sodium is a positive ion, that its movement can create an electrical gradient, and that can then attract anions or negative ions to follow. And that's exactly what happens, especially with chloride ions, which are negative anions. So the chloride ions just follow the sodium through the cells and into the peritubular capillaries, electrically attracted to the positive sodium ions that are being pumped by the sodium-potassium pump. Now, as chloride moves, as chloride is motivated to move, we also have some antiports through which it has to move. Remember, the ions don't move very well unassisted through cell membranes because they don't pass through the hydrophobic interior very well. So we do have to have carrier molecules to allow the chloride to move. As it turns out, chloride tends to move through an antiport protein in the cell membrane, apical surface, the tubular side, and it does so in exchange for other anions that they eject into the tubular fluid. So this is what prevents chloride from creating electrical imbalances. So as the chloride moves in following the sodium, other anions, other negatively charged ions, will be moved out into the tubular fluid to balance chloride's negative charge. There is also a potassium chloride symport. And I know this particular slide is a little confusing because putting the colon here, this makes you think that this is an example of the antiport that they were just talking about. And that's not the case here. This is a typo. It's a formatting typo. This right here should be listed really as a separate bullet point. It has nothing to do with what we just talked about here. So when chloride moves, a lot of it moves through an antiport that pumps other anions in the opposite direction to maintain electrical balance. But then there's also a second protein that the chloride ions can move through, which is a chloride potassium symport. And again, symport means transporting two things in the same direction. And so as chloride moves into the cuboidal cell, potassium will tend to move in as well because they share a symport molecule that has to transport both at the same time. So we have an antiport for chloride and other anions, but we have a symport for chloride ions and potassium ions. Other molecules that will diffuse, again, we just saw potassium has a symport with chloride. Potassium can also pass through the paracellular route through simple diffusion. Magnesium is reabsorbed this way, and so is phosphate, some of the phosphate. Phosphate is also co-transported with sodium ions, so phosphate is another thing that shares a symport with sodium as it moves through. Calcium is an interesting ion. Remember, calcium is very important for many body functions, and it is filtered in the glomerulus and must be reabsorbed in order to avoid dramatic calcium loss, which would ultimately lead to osteoporosis. Calcium is reabsorbed through the paracellular route in the proximal convoluted tubule, but only a small percent of calcium is reabsorbed this way. Most calcium reabsorption is going to take place in the distal convoluted tubule under the influence of hormones like parathyroid hormone, remember, and calcitriol, vitamin D, from things we learned about in AMP1. In this chapter, we're going to see a little more specifically later in the chapter exactly how those hormones affect calcium reabsorption, but that happens later in the nephron. What about glucose reabsorption? Glucose is, as we said earlier, co-transported with sodium by a symport known as the sodium glucose transport protein. Under normal conditions, all the glucose that ends up being filtered, so all the glucose that's in the tubular fluid, should be reabsorbed back into the blood by the time the fluid completes its movement through the nephron. All the glucose should be reabsorbed back into the bloodstream. So there should normally be no glucose remaining in the urine. However, if glucose levels are extremely high in the blood, as might happen, say, during diabetes mellitus, then so much glucose can wind up in the filtrate and then subsequently in the tubular fluid that it may be impossible to reabsorb all that glucose. After all, you only have a fixed number of these sodium glucose symport proteins. There are only so many of them. And if glucose concentration in the tubular fluid is so high, you might max out the ability of the proteins to transport glucose. In other words, they'll all be working as fast as they possibly can. And it's possible then that the tubular fluid might move through the proximal convoluted tubule and not get all of the glucose that was in the fluid reabsorbed. There was simply too much, and the transport proteins were maxed out. In that case, glucose might end up in the urine, or at least some of it might. And of course, that is one of the symptoms of diabetes mellitus, is glucose in the urine. This is known as glycosuria, and we have a slide that shows this a little bit later in the presentation. All right, so here we see a diagram of what we've been talking about. On the right is the lumen of the proximal convoluted tubule, so here's where the tubular fluid is. And on the left, of course, is the basement membrane and the extracellular space on the basal side of the cuboidal cell and the peritubular capillary. So as we said, sodium flow is important, and the movement of sodium into the cell along its concentration gradient drags other materials with it through symport molecules. So remember, sodium will drag glucose, it'll drag lactate, it'll drag a number of things in with it, phosphate, and so on. Remember, there's also a sodium hydrogen ion antiport, and so as sodium moves into the cell via diffusion through this protein, hydrogen ions will move out, and this is one of the ways we regulate pH of the body, is by excretion of hydrogen ions. Remember, chloride is motivated to follow sodium by electrical attraction, and chloride tends to move in exchange for other anions that will move out. And recall, there is also a chloride potassium symport molecule that moves chloride and potassium at the same time. The thing you see here that I didn't mention earlier is that this chloride potassium symport molecule tends to be on the basal side, not in the apical side. So chlorine moves into the cuboidal cell through this antiport with other anions, and then chloride moves out of the cuboidal cell into the extracellular space through this chloride potassium symport channel. And this, by the way, is one of the things that helps move potassium out of the cell, which is necessary because remember, sodium potassium pumps are always pumping potassium into the cell. And so we do want to have a way of getting that potassium back out, and that's this chloride potassium symport mechanism. Remember, water can flow freely through cells through channels called aquaporins. That's a passive movement. But water also moves through the paracellular route between the cells through simple osmosis. Notice other things that can move through the transcellular route through paracellular route between the cells through simple diffusion. Again, sodium can move, chloride chases it, potassium can move along its concentration gradient. Some calcium moves this way, though again, most calcium reabsorption is going to happen elsewhere. Phosphate can move this way, in addition to being co-transported with sodium and so on. Remember, once the sodium ends up inside the cell, we can't afford to have it accumulate here because that will disrupt concentration gradients. So we need to get it out, and of course on this side we have the sodium potassium pump that's going to pump the sodium out, and that's what drags the chloride along with it, and so on. We also have glucose pumps as well that will be pumping glucose out. And once all those things end up in the space here, the extracellular space, then they diffuse directly into the peritubular capillary, along with all the other things that diffuse through the paracellular route and wind up in this same space. All right, what about reabsorption of nitrogenous wastes specifically? Remember, nitrogenous wastes are things like urea, creatinine, uric acid, basically the results of metabolism. Well, the nephron, the entire nephron, proximal convoluted tubule through to the collecting duct, reabsorbs about half of the urea that's filtered into the tubular fluid. And some people are surprised that it's only half. So that means a fair amount of urea remains in the blood, but that's a normal, safe level of urea. The body can tolerate that much urea in the blood, so we only actually need to remove about half of it to maintain safe conditions. We do reabsorb some uric acid back into the blood, but uric acid is a bit more toxic than urea, and so most of what we reabsorb at the proximal convoluted tubule, we just secrete it later in the nephron. So we can't avoid reabsorbing it, even though we don't want to reabsorb uric acid, but we take care of that by secreting it later in the nephron. Creatinine is not reabsorbed. Remember, creatinine also is a nitrogenous waste that we do not want to have in our bloodstream, and so we don't reabsorb any of that. So that just winds up in the urine. So how much reabsorption takes place? Well, remember, on a typical day with typical glomerular filtration rates, males are producing upwards of 180 liters of filtrate a day. And yet, we're only producing, again, somewhere around 1 to 2 liters of urine. So the reabsorption rates that take place in the kidneys are in excess, usually, of 99% reabsorption, meaning of all the material, of this 180 liters that's filtered at all our glomeruli, we reabsorb a little bit more than 99% of that 180 liters. and the remaining 1% or so is what, of course, we want to get rid of, and that is what makes up the urine. Of the water that we reabsorb, 2/3 of that water is reabsorbed in the proximal convoluted tubule. And so again, 2/3 being about 66%. Earlier we said the proximal convoluted tubule does 65% of all reabsorption. So this is in line with what we've already said, about 65% of reabsorption takes place in the proximal convoluted tubule. All right, now in the proximal convoluted tubule, water, which again is largely following the active transport of sodium and then the drag of other solutes, water is reabsorbed at a constant rate. In other words, what we're saying here is that we don't have any hormonal mechanisms that will influence how much water is reabsorbed at the proximal convoluted tubule. So this is what's known, what happens in the proximal tubule is what's known as obligatory water reabsorption. Obligatory water reabsorption. There's nothing we can do about this. We must reabsorb this much water at the proximal convoluted tubule, and we don't have any hormonal or neural mechanisms for increasing or decreasing that percentage. Of course, as I hinted later in the nephron, especially in the distal convoluted tubule and the collecting duct, we will have hormonal mechanisms that will affect the final percent of water reabsorption that takes place. As we said, the peritubular capillaries are what are reabsorbing water and all the solutes that eventually wind up on the basal side of the cuboidal cells. And as I've said, this reabsorption occurs primarily by diffusion, osmosis, and solvent drag as the solutes chase the water that's moving by osmosis. Why does water move so effectively by osmosis into the peritubular capillaries? Well, a couple of things drive it. Concentration gradients are important, of course. That goes without saying, but there are some other factors that promote it as well that might not be so obvious. There is high physical pressure, hydrostatic pressure, in the interstitial space because of the accumulation of the fluid in that extracellular space. So on the basal side, on the side of the basement lamina, we continue to accumulate materials that are being reabsorbed by these cuboidal cells. That creates a physical pressure in this extracellular space, and that high pressure can tend to push materials physically into the peritubular capillaries. So that's one factor. Remember also, in the peritubular capillary itself, you've got a very low blood pressure, and that low blood pressure is due to the bottleneck created by the efferent arteriole as it drains blood out of the glomerulus. Remember, it is more narrow than the afferent. It is capable of being constricted, and so that narrowness of the efferent arteriole means low blood pressure in the peritubular capillaries. That also creates a favorable gradient compared to the high pressure in the space around it. And then finally, remember, there's a high colloid osmotic pressure in the blood inside the peritubular capillaries. Remember, colloid osmotic pressure is osmotic pressure or concentration due to proteins specifically. And the reason we have such a high colloid osmotic pressure in the blood of the peritubular capillary bed is, remember, proteins don't get filtered at the glomerulus. They're too big. And so as blood passes through the glomerulus and we lose a large amount of water into the filtrate, what's left behind is, of course, a very concentrated kind of blood, a very dehydrated kind of blood with lots of protein in it. So that blood in the peritubular capillaries got a very high osmotic pressure. And remember, osmotic pressure acts like a sponge to draw water towards it or into it. And that's also therefore going to promote the movement of water into the peritubular capillary blood, and that water then is going to drag solvents or solutes with it. And so in addition to just basic concentration gradients, we also have some of these other factors that are really promoting the movement of materials from that extracellular space around the tubules into the peritubular capillary blood. All right, now we said that angiotensin can affect water reabsorption in the proximal convoluted tubule, and that's simply what we're seeing here. Remember, when blood pressure is low, that results in the production of angiotensin II. Angiotensin II will have a direct effect on the proximal convoluted tubule, increasing water reabsorption back into the peritubular blood. That, of course, returning the water to the blood is going to increase blood volume, and that's going to compensate for the low blood pressure by increasing blood pressure. Now, earlier I said when we were talking about glucose transport that it is possible, because we have a limited number of transporter molecules, it is possible to max out the the carrier molecules and not be able to reabsorb all that we might want to. And so that's what we're talking about in this particular slide. This is just to let you know objective 106 in our chapter, the transport maximum. And so what we're saying here again is, just to reiterate, the amount of solute that can be reabsorbed is limited by how many transport carrier molecules you have embedded in those in the membranes of the microvilli of the tubular cells. Now again, it's a lot. There are a lot of these transporters because we have a huge surface area due to the microvilli. But even though there's a lot, there still is a limit. And if all those transporter molecules are working as fast as they can, then it's possible that excess solute that's in the tubular fluid might pass through the tubule and not get reabsorbed. And at that point, of course, it would end up in the urine. And so there is what we call a transport maximum for each substance. The transport maximum is the maximum amount of solute, of a particular solute, that can be transported excuse me, in a given time. And of course, again, different substances are going to have different transport maximums because they use different carrier molecules, and these different carrier molecules, there might be different numbers of them and so on, scattered throughout the cell membranes. So each solute, again, has its own transport maximum. For glucose, for instance, the one we talked about, the glucose-sodium symport, any blood glucose above 220 milligrams per deciliter of blood, that's 1/10 of a liter, is going to result in exceeding the transport maximum. Right? And so if you have this much glucose in the blood, that means you're going to have a very high concentration of glucose in the filtrate, because glucose filters very readily from blood into glomerular capsule. So you're going to have so much glucose in the tubular fluid that you will exceed the transport maximum. And so some glucose will pass through unreabsorbed, I should say, and it will wind up in the urine. And I mentioned earlier, just a few slides ago, this condition of glucose in the urine is called glycosuria. Glycosuria. Right, and this is symptomatic of several disorders, most prominently diabetes mellitus, sugar diabetes, or diabetes associated with insulin insensitivity or a lack of insulin production. And so here we see a visual of how you get to a transport maximum. Nothing new here. It's just that if you have a normal number of solute molecules, in this case glucose, if they're at a reasonable concentration, then we have enough transporters, shown in dark blue here, to transport all of those out, and none of it remains in the fluid from that point on. However, if we have an extremely high concentration of a solute in the glomerula, in the blood, we're going to have an extremely high concentration in the filtrate, which means an extremely high concentration in the tubular fluid. And all of these transport molecules will be working as fast as they can to reabsorb them. They just can't get them all, and so fluid will pass through with some of these particles unreabsorbed, and then they will wind up in the urine, again, as is the case with glucoseuria, if this particle is glucose. You'll notice on this side some of the transport molecules aren't even working, right? They're just not needed because there's just not enough solute to max out the transporters. But on this side, they're all working, and you still can't get all of the solute transported out. All right, so that takes us up through objective 108. At this point, with objective 109, we shift our focus now to tubular secretion. And remember, tubular secretion is movement in the opposite direction of reabsorption, kind of reminiscent of filtration, moving materials from the blood into the tubular fluid. But the mechanism is very different from filtration. So why do we do secretion? Again, we need to sometimes essentially finish the job of filtration that it was begun at the glomerulus. So for instance, sometimes we have to adjust the blood pH. Right? Remember, blood pH needs to be in that narrow range of 7.35 to 7.45. And in order to adjust it, sometimes we might have to secrete a little bit of hydrogen to bring the pH up, make the blood less acidic. Or we might have to secrete some bicarbonate ions to help bring the pH down a little bit, make it a little more acidic. And so this fine-tuning of blood pH by fine-tuning hydrogen and bicarbonate concentrations can be done by secretion. Notice, unlike filtration, we're talking about something very precise and very selective here, very specific. Filtration, again, just based on size, a very crude kind of movement of materials without much control. Here, we're being very specific and very precise. Secretion is also performed to do waste removal. Sometimes you don't get all of the wastes that are in the blood filtered out. You only get a certain percentage of them filtered out. Some remain in the blood as it flows into the peritubular capillary beds. And so this is a second opportunity to specifically remove some things that weren't filtered completely at the glomerulus. This includes urea, uric acid. And remember, in the proximal tubule, we do reabsorb some uric acid, so we need a second chance to get rid of it. Some acids, again, that are related to bile reabsorption in the small intestine. Ammonia, another nitrogenous waste, very toxic, so we want to make sure none of that remains in the blood. And some creatinine can be excreted. Again, this is very precise and very fine-tuned to make sure we regulate the concentration of the blood very precisely. And then, of course, if there are some unusual things in the blood that are the body considers potentially dangerous or toxic, the kidneys, along with the liver, are going to be working to excrete those or get rid of those as well. So the kidneys do work on many drugs in an effort to not just filter them out, but then secrete them as well. This, again, is the body not understanding why we're taking those drugs, just detecting them as being foreign or abnormal or potentially harmful. And examples of that include, of course, morphine, penicillin, a helpful antibiotic, and aspirin. This is why you have to take certain kinds of drugs multiple times per day to maintain a blood concentration that's therapeutic, because your kidneys are constantly working to rid the body of those, what it sees as contaminants. This is why taking certain medications can also have a cumulative harmful effect on the kidneys because of this constant stress, constant extra stress it places on the kidneys, the extra work it continually places on them as it tries to clear them from the blood. Now, we've talked about what's happening at the proximal convoluted tubule. Let's talk a little bit about what happens at the nephron loop, or loop of Henle. And we're going to be very brief at this point in the video, and we're going to come back to the loop of Henle a little bit later, talk about more specifically how these things happen. It is one of the more complicated topics in anatomy and physiology, so we're going to briefly talk about what happens now, but then we will return and talk in some detail later. So the primary function, as you see here, of the loop of Henle is to create a concentration gradient, specifically a salt gradient, in the medulla of the kidney. So what's going to happen, just again, and we want to be very brief here, and we'll return to this later, as you move through the medulla from the cortex down to the apex or papilla of the medulla, what we find is that the tissue of the medulla becomes increasingly more concentrated, increasingly more salty. So at the cortex medulla boundary, it's got a pretty typical salt concentration. But as we move down into the medulla, it gets saltier and saltier. This is what we mean by the salinity gradient. It's actually the loop of Henle that creates this salinity gradient. You may ask yourself, well, why do we even bother with this? Why do we want to create this progressively more salty tissue as we move down into the medulla? Well, to make a long story short, remember the collecting duct eventually is going to lead from the cortex, going to pick up fluid from the distal convoluted tubules of multiple nephrons and carry it down into the medulla, and eventually the urine will leak out the tips of the medulla. And so what happens is, as the collecting duct travels down into the medulla deeper and deeper, it is being surrounded by saltier and saltier tissue. Well, what is that highly salty tissue going to do? Well, it's going to have a very high osmotic pressure. And remember, that acts like a sponge to draw water out. And so this is how we're going to get the final drawing of water out of the fluid that's in the collecting duct and back into the blood. Right? And so this is water reabsorption. So essentially we're going to use this incredible concentration gradient that the loop of Henle creates to draw water out of the collecting duct as it passes through the medulla. And this is how we get that final bit of water reabsorption back into the bloodstream. And so what is this? This is water conservation. And this movement of water at the collecting duct is what's going to be under hormonal control, as we'll see in next time's video. All right, so how does it do this? Again, just as a very brief preview, and then we'll come back to this. Remember, the loop of Henle has a thick segment and a thin segment. The thick segment pulls sodium, potassium, and chloride ions. It pulls it out of the tubular fluid. The thick segment pulls these ions, active transport of these three ions, pulls it out of the fluid by active transport. It's a pump. And the potassium gets reabsorbed into the blood, but the sodium and the chloride ions remain in the tissue of the medulla. This is again what's going to make the medulla progressively more salty as you dive down into it, is all this sodium and potassium and chloride ions that are being pumped out of the tubular fluid in the loop of Henle and accumulating in the medullary tissue. Now normally you would expect water to follow this pumping of these ions, these solutes, but water doesn't follow because the thick segment of the loop of Henle is impermeable to water. And that's key, right? The fact that water doesn't follow. If water was to follow, then water would simply accumulate in the medulla as well. We wouldn't get extremely salty fluid, and the medulla would simply swell to tremendous proportions, which is not what we want. We don't want the medulla to swell with water. We simply want it to accumulate salt. And so this impermeability of the thick segment is what allows the salt to accumulate in the medullary tissue, but not the water. And so what happens is the tubular fluid, by the time it gets down to the very bottom or tip of the loop of Henle, is very dilute because we've pumped so much salt out of it, but we haven't lost any of the water. So it's a very dilute watery solution. As we'll see when we get to the thin segment, which is near the bottom of the loop and the early part of the ascending limb, that part is permeable to water, and so we're going to have lots of water movement at the very tip of the loop of Henle through the thin segment. But for reasons we'll see, that water down there is not going to disrupt the salinity of the medullary tissue because it's going to be very quickly drawn up into the bloodstream. But more on that to come later. All right, so from after we get through the loop of Henle, and we've drawn some sodium and chloride out of the fluid and drawn some water out, reabsorb some water, we get to the distal convoluted tubule. The distal convoluted tubule still contains a significant amount of water and some salts that need to be reabsorbed. And if we did no more reabsorption at this point, if this is what would become urine, we would still urinate about 36 liters of urine a day. So that's down from the 180 liters that we filter a day, but it's still clearly too much fluid loss for a person to tolerate. So we're still going to get a significant amount of reabsorption occurring in the remaining distal convoluted tubule and collecting duct. These two regions of the nephron reabsorb a variable amount of salt and water, and these are the portions, as I mentioned earlier, that are most affected by hormones in terms of exactly how much reabsorption is going to take place. Hormones that are going to affect its activity are hormones that we've talked about before, again previously in this course. Aldosterone, we know, affects sodium reabsorption and secondarily then water. Atrial natriuretic peptide, we know from prior discussion, affects sodium reabsorption in the opposite way that aldosterone does, and again then indirectly water reabsorption. Antidiuretic hormone affects water reabsorption. And one that we haven't talked too much about, we kind of hinted at in AMP1, parathyroid hormone, which affects calcium levels in the blood, is going to have an effect on calcium reabsorption here at the distal convoluted tubule and collecting duct. So we've got a lot of variable activities going on here in these two regions, and so we want to take a look at what's happening here. Here we see an electron micrograph of the distal convoluted tubule, and again, the only thing to notice is you do not see that very prominent brush border of microvilli like we saw in the proximal convoluted tubule. Okay, now there are two kinds of cells that we find in the distal convoluted tubule and the collecting ducts. The most common type are called the principal cells of the tubule. The principal cells are the ones that are lining the tubule and involved in salt and water balance, and these are the ones that have the receptors for the hormones that are going to affect the rates at which reabsorption takes place. Then we have a relatively few types of cells called intercalated cells. These are also cuboidal epithelial cells, but they serve a very specific function. The intercalated cells are specifically involved in exchanging hydrogen ions for potassium ions. And so they're specifically involved in secreting hydrogen from the blood into the tubules and in exchange reabsorbing potassium for that to maintain electrical balance. So this is yet another part of the nephron that's involved in acid-base balance for the blood. All right, so how do the hormones affect what goes on in this part of the nephron? Well, remember, aldosterone is the salt-retaining hormone. And so remember from previous discussion, it is in response to low blood pressure, for instance. Aldosterone will be stimulated and it'll be released by the adrenal cortex of the adrenal gland. As we said, a drop in blood pressure will stimulate it through the renin-angiotensin system. There are also some ways to stimulate aldosterone release more directly. So when blood sodium concentrations fall, that will directly stimulate the release of aldosterone. And of course that makes sense, right? Because aldosterone causes you to reabsorb more sodium. So if we have too little sodium in the blood, we're going to compensate for that by increasing sodium reabsorption. We can also trigger aldosterone when potassium concentrations are too high, because another effect of aldosterone is to promote potassium secretion. So again, it's a nice negative feedback mechanism. So what does aldosterone do exactly? Well, it has several mechanisms whereby it affects sodium and potassium concentrations. First, it can work on the distal convoluted tubule and the cortical portion of the collecting duct, meaning the part of the collecting duct up in the cortex, not the part in the medulla. And it also can work on the thick segment of the nephron loop. And in all cases, what it can stimulate is reabsorption of sodium, meaning it's going to pull sodium out of the tubular fluid into the surrounding space, and secretion of potassium. Remember, potassium doesn't have a whole heck of a lot of physiological significance in terms of hyper or hypokalemia. So what we can play around with potassium's levels and not worry about it quite as much as for some of the other ions. And so we do this exchange again to maintain electrical balance. Primarily that's what we're using potassium for with both hydrogen and sodium, is just maintaining electrical balance. Again, as we reabsorb sodium, pull it out of the tubular fluid and try to and get it back into the bloodstream, water is going to follow because it is a solute that it's following, and chloride will follow because of the electrical attraction. And so the net effect of aldosterone is to increase both sodium and chloride and water concentrations in the blood. It does also reduce potassium levels in the blood. That helps address, of course, the water portion, as we know, contributes to blood volume, which addresses the blood pressure issue that might have caused aldosterone to be released. It addresses the low sodium that might have caused aldosterone to be released. And it addresses the high potassium blood levels that might have caused aldosterone to be released, because it's going to bring down, as we said, blood potassium levels through secretion. We don't get as much urine produced because more of the water ends up back in the bloodstream, and the urine tends to have a higher potassium concentration because of this secretion. So these are all the effects of aldosterone. Remember, atrial natriuretic peptides are secreted by the heart in response to high blood pressure, and they have the opposite effect. They want to bring pressure down. And so natriuretic peptides are going to cause sodium to be secreted, or in other words, less sodium to be reabsorbed specifically, which means less water reabsorbed, which means less blood volume and reduction in blood pressure in response to the initial high blood pressure. So how did the natriuretic peptides do their job of decreasing sodium reabsorption? Well, it dilates the afferent arteriole and constricts the efferent arteriole. What's that going to do? That's going to increase glomerular filtration rates. And if I increase glomerular filtration rates, remember, I get faster flow of fluid through the nephron, and that faster flow reduces how much sodium and water I can reabsorb. So that's one mechanism of action. Natriuretic peptides also inhibit the renin-angiotensin system, which then is going to inhibit aldosterone. So in a sense, it antagonizes aldosterone by causing less aldosterone secretion. Natriuretic peptide also inhibits secretion of antidiuretic hormone. So remember, antidiuretic hormone is going to cause more reabsorption of water. By inhibiting it, we're going to get less reabsorption of water, which lowers blood volume, which lowers blood pressure. And then finally, it directly inhibits sodium and chloride reabsorption in the collecting duct. And so again, less sodium reabsorbed back into the blood, the opposite effect of aldosterone. And again, that means less water, and so blood pressure is going to fall again through that very familiar mechanism. Antidiuretic hormone, we've talked about before, how it stimulates the reabsorption of water in the kidneys. It is largely stimulated, its release is largely stimulated by osmolarity, rising osmolarity in the blood, meaning high solute concentrations in the blood. It happens with dehydration, things like that. This is sensed by the hypothalamus. Remember, the hypothalamic neurons are the ones that actually make antidiuretic hormone. And that ultimately triggers release of antidiuretic hormone from the posterior pituitary. Specifically, the antidiuretic hormone makes the collecting duct more permeable to water, specifically the medullary portion of the collecting duct, the part in the medulla. And we're going to get more reabsorption of water then through the collecting duct. And so we have more water in the bloodstream. That tends to then dilute the blood, which addresses the original problem of too much higher than normal osmolarity of the blood. And then finally we have parathyroid hormone, and unfortunately we're out of time for today. And so we're going to have to pick up with the hormones that affect reabsorption and secretion in the distal tubule and collecting duct. We're going to have to finish that up next time. And then we will move on to specifically how we adjust water reabsorption, once again in the distal convoluted tubule and collecting duct. So we're leaving off, we've made it up through objective 120. Next time we'll pick up with 121, talk about parathyroid hormone, and then specifically move on to reabsorption of water. So that's it for today. Stay safe, and we'll see you next time.